Hi there, my name is Jason Shoemaker. I am an assistant professor right now in the Department of Chemical Engineering, University of Pittsburgh. And I'm just going to briefly, within hopefully 30 seconds to a minute, describe some of the research we do in my lab. And hopefully we'll get to meet you all virtually when you come in and get to visit our department. So my research, and I'll share my screen so you can take a quick look at it. We do only immunology and virology pretty much in the end. So we take systems engineering tools, so tools that you'll be very familiar with from your controls engineering classes, and then try to apply those to create mathematical models to understand how does the immune response respond during infections, and then how can we improve that response. This has been a major challenge over the years, is that we kind of know pretty concretely now for the majority of respiratory infections out there, so flu and COVID-19, that your immune system often tends to be kind of a culprit behind the more severe outcomes that you observe in certain kinds of patients. And what type of patients we're talking about can vary quite a bit. We can be talking about pregnant versus non-pregnant women. We can talk about young versus elderly individuals. And even occasionally it could be something such as men versus women. Sometimes men will experience more severe disease given a select virus. Other times women will experience a more severe disease given a different virus. So these questions we want to understand is, how does the immune response play into those particular outcomes? To show a quick couple slides, here's kind of what our group looks like right now, but I need to update that slide, so I'm going to go by it really fast. Here's where we sit at, and of course right now we're living in this experience of COVID-19, so again, a lot of this doesn't really need introduction, unfortunately. But we've known for a long time, and these slides are quite old. You can see these are publications from 2008, 2009, so 10, 11 years old. We've known for a very long time that there's going to be a new virus outbreak eventually. And we've gotten lucky a lot of times. You know, we've had SARS-1 come out, which was a very deadly virus, way more deadly than what we're currently dealing with. Thankfully, it wasn't highly transmissible, and we got lucky. Now we didn't get lucky, and we can kind of see the world we're living in. The thing we want to take away, though, is these viruses are very deadly. So if we look at the right-hand side of this figure here, this is the H5N1 virus, your bird flu virus that you probably heard about in movies, and extremely deadly. More than 50% of individuals that have been known to be infected with this virus have passed away. An extremely deadly virus to handle. So you can imagine at some point in the future, if this ever becomes human transmissible, we're going to have a big problem. And what our goal in our group is try to identify ways to lower that mortality, lower the morbidity, not by targeting the virus itself, but by targeting your immune system, trying to find ways to make your immune system better adapted to respond more evenly and cleanly to the infection, so you don't have a severe inflammatory episode. Because severe inflammatory episodes are the ones that really lead to death in most cases. Of course, we can see things such as how easily viruses go global. Lots of kind of interesting tidbits and stories behind this kind of transmission studies is that, you know, we've often talked about human or animal to human transmission of disease. We don't very often talk about human to animal transmission, but you're seeing that currently in the COVID outbreak, where a lot of cats have become quite ill due to actually picking up the virus from their human owners. And we also experienced back in 2009 with the pandemic H1N1 virus, where they actually isolated in the seals, or the calcium, or what do you call these guys? Elephant seals. Back out in California, a lot of them started getting really sick. When they isolated them, I did some nasal swab testing and everything to identify what was the particular virus causing that disease. They're pretty confident is what happened was humans that were there taking pictures during tour groups up and down the California coast accidentally infected the local seal population with the pandemic H1N1 virus. So when it comes to transmission, we not only have to care about ourselves, we also have to care about the animals around us as well, because the transmission effect goes both ways. So just a quick overlook of some studies that have come out of our group recently. And what I'll try to do at the end of this video is I'll try to include a link to some of the papers here and a link to some other videos that we produced that are already on YouTube, kind of showing some of the fun things we've done in our group, including virtual reality education. But here are some projects that you could try to imagine that would be something that might be done in our group. So one thing would be using nonlinear differential equation type modeling, or nonlinear statistics in this particular case, this first project, to determine whether or not we have consistent or different immune responses when infected with a moderate virus, such as an H1N1 virus, which is a seasonal influenza, or that very deadly H5N1 virus. Was there a distinction there? And if there's a distinction, then maybe we can modulate those two responses to get a better outcome. doing the same thing here in this middle project at a slightly higher level. In the first project that we published over here, this was looking at only one aspect of immunity, that's called the interferon response, which you might have heard of in the news now. It's become kind of a big topic. Looking more broadly then at the whole innate immune system, so including things like macrophages, T cells, B cells, etc. Are those behaving differently during the course of an infection? And then another kind of project we'll do which is a little more what I would call a first principle study, is trying to understand, okay, in these really complex biological systems, to be blunt, biology is noisy, messy, disgusting. The question we always want to ask ourselves is, why does this even work? And that's one of the tools we've done, or one of the studies we've done recently, using something called agent-based modeling. You can see a nice little graphic what agent-based modeling looks like here. And I can try to jump ahead to a pretty picture of these kind of things we're running in real time. And what we've been trying to use is just try to ask questions of, if you give me a collection of cells that are getting infected, and some of these cells are really good at detecting disease and respond really quickly. Some cells don't respond very well, or don't respond at all. Why are we designed that way? What is the advantage to our body? And that was this study that was done by a student of mine who just recently graduated, Rob Gregg. And we found that you can actually make really good, robust, reliable systems by having really unreliable components, so really unreliable cells. So it was actually a pretty fun study and a pretty fun outcome. So I'm going to leave it there as the takeaway for what my group does: mathematical modeling, computational work, all of viruses, and immunology. We primarily do computational only, even though there's always the opportunity, if you want, to work in a collaborator's lab doing some basic experiments. And with that, we're just like any other lab in the world. You'll get to have the opportunity to enjoy graduate school, the opportunity to visit conferences, the opportunity to do a little bit of travel here and there. We're a pretty fun group, if I do say so myself. So if you have any questions, feel free to send me an email at any time. And again, we'll put some links here at the very end of this talk to kind of show other places that we can find information about us. Hope to see you soon.